Have you ever wondered how bacteria survive every antibiotic we throw at them? How they somehow evolve during an outbreak? Or how one small mutation in a village clinic ends up becoming a global headline? Today, we are going straight into their playbook, Bacterial Genetics. Not memorizing terms, not listing processes, just understanding how they stay ahead of us. Welcome to SHEY'S Microbiology. Let's forget the usual textbook definitions. Imagine a bacterium as a tiny startup, fast, flexible, ruthless in its strategy. At the center of the cell is its core identity, one circular chromosome, the company manual. Then floating around are plasmids, little bonus books with special tricks. Plasmids are the reason a harmless bacterium today can be a resistant nightmare tomorrow. They are like the cheat codes bacteria keep in their back pocket and share freely. This is the real secret. Bacteria are not just surviving, they are constantly updating. Every time a bacterium divides, its DNA is copied fast, and fast work means mistakes. Most of those mistakes, nothing happens. But every once in a while, a mistake creates a new advantage. Survive a new drug, grow in a place it couldn't before, switch off a target the host immune system uses. And when that advantage appears, the bacteria never forgets it. It becomes part of their identity. Evolution in microbes isn't slow. It's happening every second, everywhere. Now here's the part that makes bacterial genetics feel like science fiction. They don't wait for reproduction to exchange information. They literally share genes like files, and they do it in three bold ways. But instead of naming them like a textbook, I'm going to share it with you in a way that you will hardly forget. 1. The pickup mode. A bacterium can absorb loose DNA from the environment like picking up a USB someone dropped. If the DNA helps, it keeps it. If it's not, it discards it. 2. The virus delivery mode. A bacteriophage, a virus accidentally grabs bacterial genes and delivers them to another bacterium. It's unintentional, but it's effective. A genetic carrier service. 3. The breach mode. This is the one that spreads resistance fastest. One bacterium builds a small breach, connects to another and transfers a plasmid. No romance, just business. And once one bacterium gets that plasmid, it spreads it like wildfire. This is why outbreaks becomes harder. Bacteria don't fight alone, they help each other. Let's connect dots. Every resistance story you've heard, MRSA, ESBL, NDM, MDR-TB, all of it is genetics in motion. A mutation that changes a drug target, a plasmid that produces an enzyme, a pump that spits antibiotics back out, a pathway that rewires itself to survive. That's why resistance jumps from animals to humans, hospitals to community, one country to the world. It's not the bacteria moving, it's their genes moving. When we test bacteria in the lab, we are not just trying to grow them, we are trying to understand their genetic decisions. PCR tells us what genes they carry, AST shows how those genes behave, sequencing tells us where the strain came from, plasmid profiles explain how the outbreak spreads. Microbiology used to be about colonies and strains. Now it's about information, who carries what, who shared what, and how fast it's spreading. So that's bacterial genetics, not as a list of mechanisms but as living system. A world where microbes mutate fast, cooperate boldly, adapt instantly, and rewrite their destiny every single day. If you're enjoying this new teaching style, drop a like and subscribe. We are building microbiology into something simple, vivid, and real.